Hello, everybody. Hope everybody's having a good night. Thanks for tuning in to join us here for our COVID and neutralizing antibody webinar. Without further ado, we'll kick off here tonight. So thanks for joining. Cure Disease decided to put together an event to talk about the impact of COVID, as well as neutralizing antibodies, combining them into one topic. And we have two great speakers with us tonight. One is Dr. Federico Mingozzi of Spark Therapeutics. He's a Chief Science Officer there. And secondly, we have Dr. Noah Weisleder as well, who's a professor at Ohio State University. So today we'll start with Federico, who will be walking us through neutralizing antibodies. And then we'll hand off to Noah to talk more about COVID and COVID's impact on gene therapy. So if folks have questions, I encourage you to write them into chat. We'll try to get to as many, if there are a lot, as possible. And for those of you who are only able to stay for part of the time, we will be posting a recording of this online in the coming days. So no worries if you miss a piece. We'll have that on our replay where you'll be able to fast forward and stop. So again, if there's any questions, do use the chat feature. There's no bad question. I encourage folks to ask as many questions as they'd like. And ultimately, we want you to have a good understanding of COVID's impact, as well as the impact of neutralizing antibodies, ultimately on the emerging field of gene therapy, whether that's CRISPR or microdystrophin. It's very important, we believe, for folks to have a strong understanding, or at least an understanding of the impact of these two topics on potential gene therapies. So without further ado, I will stop sharing my screen here, and I will hand it off to Federico.
Thank you, and let me try to see. Well, that's the camera.
There you are.
And then the screen. So I'm very happy to be here tonight, and I'll try to let's see, laser pointer. Okay, so I'll try to give you a brief overview on antibodies and gene therapy. And the idea here is to provide some background and more steer a discussion rather than provide detailed information. So, just to start from gene therapy and put it into a context, when we talk about in vivo gene therapy, we typically have a vector that carries a DNA, a gene of interest, which is injected directly into the organism. And the idea is to have a single vector administration that has a therapeutic effect, and of course the long-term goal of gene therapy is safety and efficacy. So for in vivo gene therapy, to date, the most used tool to transfer a gene in a cell is derived from a virus that is found in nature. This virus that is called Adeno-associated virus. To basically transform this virus in a gene therapy tool, this was done by essentially removing the genome of the virus and replacing that with the gene of interest. There's a lot of information in this slide. I think there's 2 points that are very important. The first one is that the safety and efficacy profile of AAV vectors in the context of gene therapy, both in animal models and in humans, has been established across many studies. And the second point is also important, is the fact that AAV, the virus that was used to derive AAV vectors, naturally infect humans. And this is important because the exposure to the virus results in the formation of antibodies to the virus itself, which then can have an impact on gene therapy. I want to start really basic here, and forgive me if you know all of this, but I wanted to give some definition. The first one is around the definition of antibody. What is an antibody? And this is a very plain and basic definition. Antibody, also known as immunoglobulin. These are large proteins that basically are produced by the immune system to recognize or neutralize a pathogen like a virus. So it's really what the immune system uses to block an infection. And these antibodies have the ability to basically bind and neutralize a pathogen, including a virus, by and basically blocking the infection. The antibody titer, when in the next slides I will talk about antibody titers, I will refer to basically the measurement of how much antibodies present in an organism. And so these antibodies, of course, the higher the titer, the more is neutralizing. And neutralizing antibody is basically an antibody that blocks the biological activity of the virus, so prevents the infection of the virus. So, as I told you, humans are exposed to AAV and develop immunity. In fact, if you look at these graphs, these are antibody titers over time in humans. And you see how in younger individuals, in kids, actually, 1 year of age, in children, you don't have a lot of antibodies, but then in adults or younger children and adults, then you have a variable proportion of humans presenting antibodies to AAV as a result of exposure to the virus. And in fact, if you look, this is a survey of adult healthy volunteers. You can see that across many of the AAV vectors that we use in gene therapy, there's a variable number of humans carrying antibodies that recognize these different AAV that we call AAV serotypes. Up to 70% of the population can carry antibodies to AAV. And what is the impact of antibodies in the setting of gene transfer? I think this is basically the graphical example of what antibodies can do. So if this is the liver, the antibodies can simply block the virus from reaching the target tissue, in the case of the liver. And these are real data coming from experiments in non-human primates, where, in fact, animals that were positive, that had an antibody titer against AAV, had no effect, no expression of a given transgene. In this case, was factor 9 from the liver. And in case of animals instead that did not have any antibodies, you see how the AAV vector was able to reach the liver and express the transgene. So, of course, the impact, what I showed you earlier was a preclinical study, but what is the impact in terms of clinical trials? In AAV trials, pre-existing humoral immunity, so the presence of antibodies to AAV, is a common exclusion criterion. So basically, when you look at the enrollment in a clinical trial, oftentimes subjects that are seropositive for AAV are excluded. Of course, this is not always identical. It depends on the route of administration of the vector. There's a different tolerance for titers depending from trial to trial. And of course, there's a high variability, which is very important to point out, across studies in the kind of assay that is used to measure antibodies to AAV. So digging a little bit more in the detail, when it comes to the route of administration, so the way we administer AAV vectors, in the case of the administration, systemic administration, so through the bloodstream, the impact of antibodies on the efficiency of AAV in terms of transducing or reaching target tissues is very big. So antibodies, they have a big impact in terms of blocking gene therapy. And then, of course, there are other settings, like if you inject a vector subretinal in the eye, where antibodies don't really have an effect in terms of efficiency of gene therapy. And then, of course, as I mentioned in the previous slide, it is very important that to measure antibody, we use assays that are very sensitive and they're optimized. And in a way, the fact that across different studies, different assays are used, that makes it difficult to compare results across preclinical studies and clinical trials. Last, one other point is that it's also important, let's say, in the trial, we tend to see different threshold for antibody titers, but that is also dependent on the vector dose that is administered. And so the threshold for antibody titers may be lower when low doses of vector are used. Instead, when high vector doses are used, then there's a more, there tend to be a higher tolerance for antibody titers. So antibodies and gene therapy, a brief summary. The antibodies to AAV are commonly found in humans, and they have an impact on the efficacy of gene therapy. In fact, AAV vectors may be neutralized and therefore not efficient neutralized by antibodies and not efficient in reaching a target tissue. The impact of antibodies depends on several factors, as I mentioned, including the route of delivery of the AAV vectors and the total dose that is given. And again, it's very important to measure antibodies with sensitive assays. And so again, so far I discussed mostly what happened before vector administration, right? I told you that humans are exposed to AAV, adeno-associated virus, and therefore develop antibodies. And so we need to measure them before we do we enroll into a gene therapy study. But what about after? What happened after gene transfer, so after the administration of an AAV vector? And then, based on that, can we actually get to give multiple doses of AAV vectors? The answer is in these next two slides. This is, in fact, published data from a clinical trial where an AAV vector was given at the time 0 here, where it was shown here with a green arrow. And as you can see, after gene therapy, I think you can probably follow the orange line is the best one. I mean, it's the most representative. Let's say these are the IgG, where you see the antibodies develop. And this is a follow-up of up to 100 days. And then after, we also recently published data in which we showed that up to, in this table, you can see that up to 16 years after gene transfer, antibodies to AAV persist. So they remain stable over time. And so essentially, this, and by the way, these titers are quite high, higher than the ones we find in nature, and they persist, as I told you. And the implication is that doing having a re-administration, so a subsequent administration of an AAV vector after the first administration becomes very difficult. This is about induced humoral immunity, so induced antibodies to AAV. The administration of AAV vectors triggers the production of high-level neutralizing antibodies, which tend to persist at high titers for several years post-gene transfer. And as I told you, currently it is not possible to administer multiple doses of AAV vectors. Of course, we are also thinking about solutions. So in the next few slides, and then I will close, I will try to answer the question, which is important: what can we do to overcome anti-capsid antibodies? And again, the goals here are to expand eligibility, so address the natural immunity to AAV, and also be able to dose multiple times AAV vectors. If you look at the literature, there's a lot of work published. And so I don't want to go into detail. These are some examples of using, for example, pharmacological immunosuppression. I will talk about the removal of antibodies, or even there's ways to modify AAVs to make them less neutralized by or more resistant to antibodies. In the case, I'll give you just two examples. The example one is the one of plasmapheresis, which is a technology that is used in the clinic, is available in many hospitals, and basically consists in the use of a column to remove immunoglobulins, so antibodies, from the circulation, and the plasma is simply run through a cycle and then infused into a subject, a patient. So this is a technology they use, for example, in autoimmunity in several diseases. So it is available in the clinic. And several groups look at this technology in the context of AAV. And for example, this is an older study published now 10 years ago, where they basically look at what happened to antibodies to AAV before and after, in this case, 4 cycles of plasmapheresis. And I just want to make you follow, I'll help you follow the numbers. Basically, as you see, the numbers in this column, the before the first cycle of plasmapheresis, are definitely in some cases elevated. And then after 4 cycles, there's a significant drop in antibody titers. And so this could be a solution to remove from the circulation antibodies. The second example is an example that we published recently, which is the use of an enzyme to basically digest antibodies in plasma, and so allow for the vector to get to the tissue of interest. This enzyme is actually the interesting part about this enzyme, which is called IDS, and is the fact that this enzyme is able to cut antibodies, so cleave antibodies, therefore, which then are cleared. And in fact, this is an enzyme that is currently tested, and actually in Europe is approved, but in the US is still being tested in the clinic in the context of transplant, kidney transplant. And from this slide, this is from a manuscript that was published in 2017. You can see that after the infusion of this enzyme IDS, there is a complete disappearance of antibodies, all antibodies from the circulation, which then reappear in a matter of a couple of days. And so can you actually do that the same in the context of gene therapy? And again, last year we published a study where, in fact, we use this enzyme to essentially enable the repeated administration of an AAV vector. And this is the study design. These were non-human primates that received first a vector, time 0, then 210 days later, received the pretreatment with the enzyme, and then again a second vector to demonstrate that we were able to re-administer AAV. And so by doing that, we were able to show that the treatment with this IDS enzyme resulted in a decrease in antibodies to AAV, and also resulted in essentially enable the re-administration of the AAV vector the second time around. So this is a short presentation. As I say, I hope gave you a little bit of background, and probably and allow you also to ask questions. The summary, I think, is pretty obvious. As I told you earlier, AAV antibodies are highly prevalent in humans, and are a limitation in terms of patient eligibility and ability to redose AAV vectors if needed. So there's many technologies now being explored. I gave you a couple of examples, but the field is really working hard to find solutions to the problem. And hopefully we'll have them. We'll have some clinical data to show you at some point soon. I'm going to stop here. Thank you for your attention, and then I can perhaps answer questions.
Thanks so much for the presentation, Federico. A couple questions that we had. Here, let me join you so you don't feel like you're alone. A couple questions that we had come in. I'm just going to start from the bottom. Good questions. The first is, what's the epitope similarity among different AAV serotypes if an individual has neutralizing antibodies? And I'm assuming that means significant levels.
That's a great question, yes. So The AAV tend to be very conserved, both in terms of amino acid sequence and structure, and so you do have a high degree of, let's say, B-cell epitope similarity and cross-reactivity. So we do see a lot of cross-neutralization.
And the second question is there any relationship between the dose-dependent neutralizing antibody development and underlying immune profile of the patients prior to vector injection, such as B and T cell profile? Any thoughts on that?
Question. However, I don't think there's enough data that will really point out to any background in terms of immune system, genetic background that would lead to higher or lower antibodies. Probably there are some factors, but they have not been studied.
Yeah, really interesting question. I agree. One of the things that we're starting to find in our early research study, looking at neutralizing antibodies across several different serotypes, is that for instance, we analyzed a pair of brothers, one of whom was older yet had lower neutralizing antibody levels against the same serotypes versus a younger brother who had higher levels than the older. We thought that was really fascinating.
Yeah, I think that's a very good point. There's and I would say I think probably that will be something that we should probably pay attention and try to study. But it's true that there's not a lot of consistencies. In fact, even if you look at antibodies and maybe the T cells, the other arm of immunity, they don't really correlate, and so we don't quite have an explanation for that, but it is something that we've seen as well.
Yeah, super interesting. The next question was asked by Jose. How is that in animal studies? I think this was in reference to your 4th or 5th slide early on, if I'm not mistaken.
So I showed you so all the data about removal antibodies, let's say, they were basically preclinical in a way, because even the study on plasmapheresis, those were samples coming from patients that were treated with plasmapheresis not for gene therapy. It was for autoimmune indication. So those are all preclinical, and the last study that I showed you were monkeys. And then I believe before I showed you mostly actually data from either clinical trials or healthy donors.
Thank you. And that last question, we'll hold that one until the question is, which we can hold until after Noah's presentation, I think. Are any of the current or upcoming COVID vaccines going to be using AAV? And if so, will patients who receive these types of COVID vaccines not be eligible for gene therapy trials in the future that use AAV? Are these technologies you describe a possible solution to this problem? So a couple questions baked in there. I don't know if you want to tackle this after Noah's presentation or if you want to give a few words now, but whatever is more comfortable for you.
I can. It's the same for me, meaning that I think these are great questions. Most of the vaccines, actually all the vaccines approved to date, they use technologies. They're not based on AAV, therefore it's unlikely they will interfere with gene therapy. Of course, there's way to test. As you know, we can measure antibodies and we can actually assess that, but it's unlikely that a vaccine of the ones that have been approved so far would generate antibodies against AAV that we use in gene therapy.
Thank you. What a beautiful segue we have here for our next wonderful speaker here. Thank you, Federico. We'll come back potentially towards the end for Q&A, but thank you so much for your great presentation.
Thank you.
Without further ado, we'll move on to our esteemed professor, Noah Weisleder, who is a professor at Ohio State University. Noah will be taking us through insights into the COVID vaccine development as it relates to the topic at hand. So without further ado, Noah, all yours.
Thank you. I appreciate the introduction there. And certainly also a wonderful presentation by Federico there that'll, and I echo his last statement there. He said it much more succinctly than I will. I'll take 15 minutes to say what he did in about 15 seconds, but hopefully it'll still be illuminating as we go through here. Because what I'm going to talk about is exactly that question that we just had at the end. So a lot of people on today's presentation are interested in knowing, with all of the vaccines that are coming out, does this affect your the likelihood that you'll develop these antibodies against AAV, and that might affect the efficacy or status of your ability to be able to have effective gene therapies as they become available. So that's what I'm going to talk about today. I'm going to walk you a little bit through the COVID-19 disease and its accompanying virus, and but then talk a little bit more about the viruses that are used for gene therapy. I don't have to talk too much about those because we've already had an excellent presentation on those. And then just basically answer the general question: how much do you have to worry about that? And to give the spoiler away, actually not too much, but there is one specific example that I certainly want to make sure you're aware of. So we'll continue on here. So just to start, I mean, when I work with people in my group and people and collaborators, any project I'm working on, I always like to clearly define what the challenge is at the beginning. So we'll spend a moment here clarifying exactly what we're looking at. So a couple things to say here, just terminology-wise. So we always hear about COVID-19. You hear it on the news every day. However, really, COVID-19 officially refers to the disease that we're talking about, that which happens from an infection with a particular novel coronavirus, and that's the SARS CoV-2 virus, which was originated in Wuhan, China, and has obviously become a pandemic throughout the world. And what this virus really is we see it over here in this electron micrograph. That's what COVID really looks like. If you get it under a powerful enough microscope, you can see the individual viral particles. And this is 3 of them here. And what you see out there is the spike protein you've heard about. That spike protein is what most of these vaccines against the COVID-19 vaccines against SARS CoV-2, that's what it's targeting. And really, the SARS is quite a large virus. It's almost 100 nanometers across, so that's very large for a virus. It's very small for many things, but for a virus, that's quite large, and we'll talk a little bit more about that later. And it's one of these class 4 coronaviruses. That's not important, but it does mean that it's going to be contagious to humans, and that's where the real problem is here. Coronaviruses are endemic in most mammalian species, and we have many of them. Many cold viruses are coronaviruses. But what really causes the problem when we're talking about this coronavirus is that it's novel and no one ever been exposed to it before. And that it spreads fairly easily relative to some coronaviruses between people through close contact or through respiratory droplets. And that means that it very easily can move from one human to another. And thus, that's why we have such a challenge here for dealing with this disease. It's an interesting virus too, in that it enters the cell through a angiotensin converting enzyme receptor. That's important because a lot of these different, that's actually why you see such an effect on the lungs and in the cardiovascular system, because that's a common way to control both your lungs and your heart is the angiotensin system. So that's one of the reasons why you see a lot of lung complications and a lot of cardiovascular complications that come from these from infection with SARS. And then once it gets in those cells, it can hijack those cells and make copies of itself. That's just a little summary of how the disease works there. There's certainly more we could go into, but that's where that's there. I thought I saw, did I see a question come up? No, okay. So now you saw that picture we showed you before. That's an electron micrograph. What we have here is a sort of a colorized version, sort of an idealized model of what the SARS CoV-2 virus particle looks like. And as I mentioned before, this is a large virus, and it has a really large genome. And this kind of contrasts with what you saw earlier in Federico's presentation with Adeno-associated virus, or AAV. We'll see that in just a minute, a very similar figure. But this is a large virus with many open reading frames. It makes a lot of different types of proteins. it's highlighted here is 3 of the key important ones that you see here, and ones that we have structures for already. I've never seen as much progress on something as quickly as you see from SARS. But this spike protein is coded by a portion of what is the genome of this particular virus, and that's what, as I mentioned earlier, most of these vaccines are going to target. And that's because if you look at the virus over here, it's shown in red. All these little red knobs that stick off of the virus are those spike proteins. So it's very clearly presented out there, and it makes it easy for the body to recognize those particular spike proteins and to generate antibodies against them. So that's why that's what you use in your vaccines, because that's what the body is going to most likely generate antibodies for, and be able to target and treat internally, be able to treat this SARS virus. Now, you can contrast that with the virus we're interested in today, and that's the adeno-associated virus. And why is that of interest? Because it's used widely for a lot of different types of either on the market or soon to be on the market, hopefully, gene therapy treatments. And so we already saw some of these maps here, so we don't have to go into a lot of the detail on it. But you can contrast in several ways how this adeno-associated virus is compared to the COVID virus that we just talked about. So these adeno-associated viruses are very small, actually, even small for a virus. They're only 20 nanometers across. So the COVID virus is 5 times larger than the AAV virus that we see here. And it also contrasted in that, as we discussed before, it doesn't cause any known diseases in humans. It can infect their cells, but it doesn't actually cause a disease phenotype. So that's an advantage too. And as a result, it's really become the platform that almost all gene therapy treatments that are coming out use this as a delivery platform. But if you look at its genome, you can see that it really has no relation or homology to this SARS CoV-2 virus that causes COVID-19. It's a very small genome, relatively speaking. We're about 5 kb versus the 30 kb you see in the SARS virus. So again, that's almost 6 times the size of the genomes that they are. And there's only the 2 genes that you find within your AAV, which are usually removed in order to have the cargo that they're going to deliver for gene therapy. So this gives you a good idea about the difference in the type of virus that we're talking about here. AAV is very different than what you see in your from your SARS CoV-2 virus. So, and I'm going to talk about a third virus here, and that's important for a couple different reasons, and I'll talk about those as we move on here too. But this is somewhere that's a virus that's about in between the AAV and the COVID-19 virus. So this is an adenovirus, which I refer to as AV, and some of you may be familiar with this too. This was one of the first viruses that was actually intended to be used for gene therapy. It was developed as gene therapies, but then was eventually abandoned for the superior, for several reasons, AAV. However, it is important to keep this in mind, because this is, while it's not being used for gene therapy approaches to treat inherited diseases, there are several vaccines that are developing the adenovirus as a way to deliver genetic material from the COVID virus, so a virus delivering a virus, as it were, in order to produce an immune response and act as a vaccine against COVID-19. However, there's still no relation or homology between either AAV or the SARS CoV-2 vaccine, between the adenovirus, between the AAV, or the SARS CoV-2 that causes COVID-19. And you can really kind of see that if you bring this up on this next slide here. So these are these 3 images I showed you before, with showing these difference in sizes and a difference in structure. So you can see that these are very different viruses. The SARS is the largest of all of them, has that prominent spike protein that appears throughout the external coat of the virus. The adenovirus is slightly smaller, and it has other proteins that are present on its external coat as well. However, they're less dense. and it's much different. It's a much longer but less protein that doesn't bond the same receptor. And then your adeno-associated virus is even smaller, with a more dense but much smaller protein that's involved in the actual capsule that surrounds the genetic material. So you can see that these are very different viruses. And as we talked about earlier, the way the immune system recognizes viruses is going to target principally those exposed proteins. And you can see how different they are as you go across these different viruses. So as a result, when the body sees these, it's going to recognize one specifically, and it shouldn't generate antibodies against the other. So if you have a SARS vaccine that's going to target this particular virus, there's going to be very little chance that it's going to affect the AAV virus or the adenovirus, because they're very different viruses. One way to think about these viruses is to think about them how different they are from one another, right? So if you think about the adeno-associated virus, and we make an analogy of that to, I seem to be having some minor technical problems here. We seem to have skipped forward. There we go. Okay. It's moving very slow here, but yeah. But if you want to make an analogy, if we were going to talk about mammals, right? So if an adenovirus is very small, we could think of this as a mouse. We think of adenovirus as something larger, considerably larger, perhaps a horse. And this SARS virus would be something like a blue whale. You know, they're all mammals, but they're all very different. So when you look at those three different animals, you won't see you'll see a mouse versus a horse versus a blue whale. And then your immune system will think about it in the same way. It's going to recognize the SARS virus as something very different. Then it's going to recognize the adeno-associated virus. So the response mounted against the SARS virus will not affect the adenovirus, and actually vice versa. There we go. So you can see from there, there's a distinct difference between these viruses, and the immune system is going to treat them differently. So that the lack of homology, the lack of similarity between these different viruses means that the antibodies produced by almost all of the SARS CoV-2 vaccines that will be available are not going to cause any they won't recognize the AAV, they won't recognize the adenovirus either. And as a result, virtually there really isn't any concern that would be in having a vaccine against this SARS CoV-2 virus, and how that there's no concern that would affect your AAV serotype sensitivity, right? So, however, as with most things in biology, there is an exception to that. So there's at least one exception to that you should know. And that exception is the vaccine that's in development called AAV COVID, which is a COVID-19 vaccine. So this is actually an experimental vaccine. It has not gotten into clinical trials yet, but it's actually very close to doing that. And this is a gene therapy-based delivery where they're using an adeno-associated virus to transfer a portion of that the genome coding for the spike protein in the COVID-19 virus into a patient in order to be able to produce an immune response. So you've heard about the Pfizer vaccine that's out there and the Moderna vaccine that's out there. Those use lipid nanoparticle to deliver that, whereas this vaccine would use an adeno-associated virus to do that. This is coming from researchers at the Massachusetts Eye and Ear Institute, and they're working with a series of partners to do that at the University of Pennsylvania, a bunch of contract manufacturing groups, as well as a Novartis subsidiary of Exis. These are all partners that are working with them to develop this vaccine. So as I mentioned, it's not close to being on the market yet, although as you know, these things are moving quite rapidly. So this is something you probably should be concerned about if you want to be concerned about your AAV antibody sensitivity and titer that you might have. Because this is a vaccine that will be delivered by AAV, so this would produce an immune response that's likely to affect your AAV state. The AAV that they're using is actually one that is not used, as far as I know, in developing any gene therapy that's coming on the market. It's a rhesus monkey serotype that was developed in Jim Wilson's lab over 10 years ago. They actually, when they developed it, it's because it comes from a rhesus monkey. There's actually less antibodies about that in the general population than you would see generally, which is one of the reasons they were looking at these, for looking at ones that might not produce much of that don't have much of an immune response to them already. However, even though there's not much of a native immune response present to that, as we talked about earlier, the different AAV serotypes are very similar to one another. And with them being very similar to another, they have a similar structure. And with a similar structure, that means that if you were to use this AAV COVID vaccine, then you would likely have a response that would affect other AAV serotypes as well, and might have an effect on different types of gene therapies. So it's probably likely that you're better off to avoid any SARS COVID vaccine that is making use of an AAV serotype for delivery. As far as I know, and as far as I could find out from talking to a number of people, just because there's a lot going on in this field, is that this is the only vaccine that's currently using an AAV serotype as delivery. So every other vaccine that's being developed by COVID-19 would have no effect on your AAV antibody status. The only one that you need to be worried about is this COVID-19 AAV COVID vaccine, which is not on the market yet, but would be marketed in this similar fashion, so you would be aware if this is the vaccine you're working with. Every other vaccine would be perfectly safe to use, and you would avoid having any sort of effect on your AAV antibodies. That's the only one you would need to avoid at this point. I'd also like to point out, just to make sure that everybody knows, that Novartis is working on a lot of other things related to COVID-19. So just because you see something from Novartis that is a treatment for COVID-19, that does not mean that it will affect your AAV status. That's because they're working, for example, Novartis is manufacturing some of the Pfizer vaccine. They're looking at 2 of their existing drugs to see if they might have efficacy against COVID-19. They also have collaborators who are developing new therapies that might have an effect. So all of these do not involve AAV, and they will not affect the status of the AAV antibodies in patients. So these other types of treatments for COVID-19 are also perfectly fine to use. They will not affect your AAV serotype at all, and most of these don't involve any sort of gene delivery mechanism anyway. The reason I brought up adenovirus is there are multiple vaccines that use adenovirus, AV, to deliver as a gene delivery mechanism. Those are also perfectly safe for people who are concerned about their AAV status, antibody status to use. AAV and AAV, even though they sound very similar, are, as we talked about earlier, very different. That was the mouse versus the horse. So that would, so under those conditions, you don't need to worry about AAV causing you to get antibodies against AAV. So any vaccine that makes use of AAV adenovirus will also be safe. It is only that one vaccine that's using adeno-associated virus you have to be concerned about. So just to summarize, there's no relation or homology between AAV and either the SARS CoV-2 virus or that causes COVID-19, or adenovirus either, for that matter. So the fact that there's no homology, there's no similarity, means that antibodies produced by most of the SARS CoV-2 vaccines, they're not going to recognize adeno-associated virus or adenovirus. So you're perfectly fine using any vaccine there should not be an effect on your adeno-associated virus status. However, you are better off avoiding any SARS-CoV-2 vaccine that makes use of an AAV serotype. Currently, that's really just that AAV COVID vaccine that's being developed. Other treatments from companies that are making that would be perfectly safe. And any vaccine using adenovirus would also not affect your AAV antibody levels. So that's the presentation that I brought along. I'm happy to take any other questions that you might have. I couldn't see them scrolling up there, so I don't know if Rich has any, or please feel free to put them in now.
Cool, thanks, Noah. One question we got was whether the difference in an RNA or DNA-based vaccine will have any impact on proposed immunogenicity or expected.
Well, I mean, I guess it depends on what you mean by immunogenicity, but
to AAV, sorry, to AAV neutralizing antibodies.
That should not matter. So basically, most vaccines are going to be raised against protein, right? So the old central dogma of molecular biology, which probably most of you don't know, but we drill into our students over and over again, are that DNA makes RNA, which makes proteins. So you can make so you can either put in DNA or RNA, and both will end up eventually making proteins. And that's really what is being recognized by the immune system in this case, is the protein that's developed. So it shouldn't make that much of a difference whether you put in DNA or RNA. Now, you saw that as we went through. The COVID virus is a RNA-based virus, whereas the AAV and AV are DNA-based viruses. But that's still not going to make much of a difference in terms of how there's an immune response that takes place. So that's not really a concern. Either a DNA or a RNA-based vaccine against COVID-19 is not going to have much of an effect either way, because they'll be focusing on the proteins that are produced.
Well, thank you so much. I learned a lot from your presentation. Really appreciate it. I think all the folks listening did as well. There was one question that just popped in. Is there any risk in blood transfusion if the donor has neutralizing antibodies against the COVID vaccine? I'm not sure I quite follow that question. Is there any risk in blood transfusion? Maybe it's any risk of blood transfusion if the donor has neutralizing antibodies against COVID vaccine. Federico.
I think the question is really if you can transfer antibodies, if there is any risk also. I think there's also a mention of AAV antibodies.
Right. Yeah, so I think that, I mean, my interpretation of that question, and of course, if the if the question asker wants to follow up, please feel free. But my interpretation of that question is, should you be concerned that you'd be bringing vaccine, that you'd be bringing some sort of bringing some sort of antibody, or it's close to my bedtime. Sorry, people. Bringing either an antibody or a virus into you that might cause an issue. So if that's the case, so actually that's an interesting question, because at least in Ohio, I can't speak for every state, but I know that all blood transfusions in Ohio, we've been screening them for the presence of antibodies against AAV, not against AAV, against COVID, because there's been multiple hospitals that actually use the serum isolated from those blood products that do show antibodies as a treatment for treating the patients who have COVID-19. It's called convalescent plasma treatment. So that actual approach is it's less used now that more sort of treatments are coming online, but we're still testing for that. So a blood transfusion, the blood being transfused, at least in Ohio, and I would imagine that's pretty standard in all the country now, we'll know if those antibodies exist. Now, I wouldn't be too concerned. Oh, please go ahead.
No, please go ahead.
Yeah, I was going to say, I wouldn't have too much concern about that, because you're not transferring the cells that produce antibodies at that point, right? So the most you would transfer is the antibodies, and the antibodies themselves would have a limited shelf life. Basically, they turn over in the blood eventually, and you wouldn't have that over an extended period. So I don't think there'd be a big risk to that. And the idea of the viruses being transferred is very unlikely as well. Blood products are heavily screened for many viruses. Again, that varies state to state. But I'm sure that there would be screening. Well, there'd certainly be screening for most viruses. The lipid particles, they have one of the advantages of that of the lipid particle vaccines is they have a very short half-life. That means they go away very quickly in the blood. That's very good from a safety point of view. So you wouldn't have that take place. So I don't think there'd be a lot of risks from having a blood transfusion. And basically, bottom line, if you need a blood transfusion, you're better off having it in most cases.
Great, thank you so much. So I think that's the end of the questions. If there's any last minute questions, do shoot them over now. But I just want to give you both a big round of applause for taking time out of your busy schedule to talk with our group tonight. On behalf of Cure Rare Disease, just want to say that I really appreciate it, and we really appreciate it. I know our folks who have joined tonight appreciate it as well. So a big thank you to you both.
Thank you.
It's a pleasure to be here.
Yes.
Right. All right, so that's a wrap for us. If we will, like I mentioned, we'll put a recording of this out on the website for folks to tune into later on, if they so please. But thank you so much for everybody joining tonight, and stay safe out there. Thanks a lot.
Have a good evening.
Bye. Thank you.
Thanks, Rich. Let me know if you need anything else.
Will do. Thanks, Noah. Have a great night. Bye-bye.